These are the Chapter 5 Notes 4 on experiments and completely randomized design. Today we're going to talk about placebos and the placebo effect. We're also going to look at some vocabulary associated with running experiments. We're going to talk about the 3 characteristics that make up completely randomized design, and you'll learn how to outline an experiment as well. The first thing I want to talk about is this first question on your note sheet. How can you determine if research is an experiment and not an observational study? It's kind of important to understand the definition for experiment, which we talked about the other day in class. And if you forget it, remember, it appears on your calculator. This is a TI calculator. TI are the initials for what makes up an experiment. In an experiment, a treatment is imposed. So how can you determine if research is an experiment? If a treatment is imposed. Now, when you run experiments, we oftentimes use something that's called a placebo, and what could possibly result is something called the placebo effect. Many of you watched a short video on what placebos and the placebo effect are, and we'll take a look at these definitions here in just a moment.
Now, you've all heard of the placebo effect before, when something with no known therapeutic value can actually make people feel better. It's a great trick our minds play on us, that by believing and expecting something to work, it actually does. But what's weird is that the strength of the effect can differ for some really strange reasons. For example, the same placebo can treat pain half as well as aspirin, while at the same time treating pain half as well as morphine. Morphine's a much more powerful painkiller, but a placebo is half as effective as both. Saying a placebo will reduce pain reduces pain, but saying that the same placebo will increase pain increases pain. Believing that a placebo will make you feel better will make you feel better. Believing that it won't has the opposite effect. Now, placebos aren't just pills. They can be creams, injections, surgeries, or drinks. You can even get placebo buttons. They don't actually do anything, but they shall make you feel like you're in control. But not all placebos are equal. The effect of the placebo is bigger when the pill itself is bigger, or if you have 2 instead of 1. Or 2 once a day instead of 1 twice a day. And a capsule will usually beat a pill, and a syringe will usually beat a capsule, and anything with a science machine can outperform any of them. A plain pill works worse than a branded one. A discounted pill works worse than a pricey one, and even a pill in a plain box does worse than one that's all shiny. Placebos that are blue work best as downers, and placebos that are red are better as uppers. Studies have shown that people who take their meds on a regular basis are less likely to die than those that don't, even if those meds are all placebos. You can even get addicted to placebos. In one study, a group of women took placebos for more than 5 years. 40% of them suffered withdrawals afterwards. In fact, the effect of placebos can be so strong that some people want them banned from sports. But I mean, how would you even test for that? Placebos don't even seem to work from place to place. For example, in Germany, using a placebo to treat ulcers works better than anywhere else in Europe. But using a placebo that treats hypertension doesn't work nearly as well as it does for its neighbours. Now remember that all of this is about comparing things that both have nothing medically effective in them, which goes to show that a placebo isn't about what's in it, but about the beliefs that we load onto it. Our minds create the medicine, and that is pretty freaking weird.
Alright, so good stuff on that video. So to recap, what exactly is a placebo? Well, a placebo is anything that seems like a real treatment, but isn't. And you might be wondering, well, why would anybody even volunteer to take a placebo in the first place? Well, typically, if you're thinking about a medical study, usually placebos are used with some sort of experimental treatment. So let's say you're trying to Okay, that was bizarre. It's like night here and all the lights turned off in my room all of a sudden. Anyway, I'm cool, so here we go again. Why would you take a placebo? Well, usually placebos are done as part of an experimental study or research on some new medicine that they want to try out. And to show that the medicine actually works, they'll ask for volunteers to be part of a study. And they'll say, okay, Some people in this group will receive a placebo. Some group of people will receive the actual medicine. We're not going to tell you what you're going to receive, but if you volunteer to be part of this study, maybe you earn a little bit of a stipend, or maybe if you're suffering with some sort of ailment, you might be able to find some sort of relief. So placebos are not something that people voluntarily take. A placebo is something that you're not aware is actually not medicine. So anything that seems like a real treatment but isn't, that qualifies as a placebo. The placebo effect occurs when a person has a positive or a negative response to a placebo. And again, the video that you just watched showed you a number of different cases where the placebo effect is certainly in action. And again, people respond differently depending on the area of the world you live in, what color the pills are. There's all sorts of interesting things that happen with this placebo effect. We're going to next look at some vocabulary that's associated with experiments. We're going to take a look at an example of an experiment where we have imposed a treatment. In this case, we're going to look at a taste test on cookies with differing salt levels and differing cooking temperatures. So we're going to cook 3 cookies at each of these different salt and temperature readings. So we're going to make 3 cookies in which the recipe used 1 teaspoon of salt and the cookies were cooked at 300 degrees. We'll bake another 3 at 350 degrees with the 1 teaspoon of salt and 3 more cookies with 1 teaspoon of salt in the recipe cooked at 400. We'll do the same 3 cookies at these temperatures with 2 teaspoons of salt as well. So there's kind of 6 different batches of 3 cookies. Each set of 3 cookies has a different salt content and was cooked at a different temperature. In this situation here, the units in statistics are the individuals on which an experiment was performed. A lot of times when we conduct research, it's done on humans or lab animals or something of that nature. In this problem, our units are actually the cookies. We're performing an experiment on the ingredients on the cookies. If you just want to say the cookies, that would be acceptable. So when we're done, we're going to have cookies that have different salt contents and they're cooked at different temperatures, so we may get kind of different types of cookies. The units in this problem are the cookies. Now, we call the units subjects if the units are humans. So subjects are human units. Obviously, in this case, the cookies are not subjects because they're not humans, even if we give them arms and legs. Factors. Factors are the explanatory variables in the experiment. So factors and explanatory variables are talking about the exact same thing. If somebody goes, why do the cookies taste the way they do? You would probably in real life say, well, there's many factors. We looked at 2, temperature and salt content. Those are your explanatory variables. So we call them the factors as well. This experiment had 2 factors. Some experiments only have 1. Levels. The levels are the specific values for each factor. We have 2 factors, so we're going to look at the levels for the 2 different factors we have. For salt, there are 2 levels, 1 teaspoon and 2 teaspoons. For temperature, there are 3 levels. These levels are the different temperatures, 300 degrees, 350 degrees, 400 degrees. Those are your levels. The treatments are all combinations of these levels. If you have multiple factors, it's nice making a table like this because it sort of diagrams out how many different treatments there are all together. Because we have 3 temperatures and 2 different salt contents, you'll notice that there are a total of 6 treatments in this case. Another way of laying out the treatments is to make a little tree diagram. If you want to go, one of the treatments is 1 teaspoon at each of these 3 different temperatures, 300, 350, and 400. Those are 3 of the treatments. And the other 3 treatments were the 2 teaspoons of salt at these 3 temperatures. So all of the combinations of the levels of the factors make up the treatments. Easy way to do it is to multiply the levels for each of these different factors we're working with. As we move to the back side of your notes worksheet, we're going to look at something called completely randomized design. An ideal experiment utilizes something called completely randomized design, and this has three different elements that you want to talk about here. The first element we're going to look at is something called control. Now, control doesn't necessarily mean a control group. Control means you've got control of your experiment. Your goal is to keep extraneous factors constant. So in the case of the cookies example, what that might mean is we're going to adjust the salt contents, we're going to adjust the temperature, but we're going to otherwise use the same amounts of batter, we're going to use the same ovens, we're going to keep all of these other extraneous factors constant. Why is that important? This reduces the effect of confounding variables. Going back to the cookie example, if you want to show that salt content has a negative impact on taste, like the more salt you add, the worse it tastes, what you don't also want to do is you don't want to accidentally leave some of the cookies in the oven for a longer period of time than others. So you don't want somebody going, well, yeah, you got that research, but there were these other factors that fit in here. We want to control these confounding variables. We want to basically not let them be part of the experiment. We want to keep these extraneous factors constant, so really your experiment is solely focusing on these things that we are changing. The second thing that we're going to do is we're going to use randomization. Our samples are going to be taken randomly or assigned randomly. Why do we want to do this? Well, we've been talking about this recently in class. The idea that when you randomize your sample, when you don't rig the sample, load it up with people that you think may respond favorably, it makes your research better because it reduces bias. That is the sole purpose of randomization, is that it reduces biased results. If you select your sample randomly, somebody can't come back and go, well, you selected these people because you knew they were going to do well. Nope, we selected them randomly. That takes away the argument that you're rigging your data, and it provides kind of that element of fairness, which is nice. The final element of completely randomized design is something called replication. Replication involves repeating an experiment many times or using a large sample size in your experiment. So why is this important? Take an example of flipping a coin. It's very possible if you did an experiment with coin flipping, if you flipped a coin twice, it wouldn't be unusual to get heads 100% of the time. If we repeat an experiment many times, it's really unlikely that we'd get 100% if we flip, for example, 100 coins. So if you're talking about percentage of coins that land heads or tails, it's probably a good idea to engage a large sample size. Similarly, by repeating an experiment many times, maybe if you only ran an experiment once, you got goofy data. It was one of those full moon instances where, whoa, I wasn't expecting these types of results, and it's what we got. If we repeat an experiment many times, we may find that that first time was kind of an anomaly and that we got better data as we increase the number of times we ran the experiment. So replication is a good way to reduce what's called chance variation. Chance variation is that kind of once in a great while goofy answer that you possibly could get if you run an experiment maybe once or use a small sample size. So if we repeat an experiment many times, it's likely that we could go, ooh, this goofy result is not really typical of what we typically would see. So because we ran the experiment many times, we found that was more of an anomaly than anything else. The last thing we're going to address is how to outline an experiment. Now, an outline for an experiment is a visual way of displaying completely randomized design. In an outline, you always start with random assignment. Then you include a branch for each group. You're going to number the groups and give a size and label for each group. Usually group 1 and group 2 when you talk about how many items or units or subjects are in each group. You're going to describe the two treatments. You're going to label them treatment 1 and treatment 2. If a treatment's placebo, you can call it that. And at the end of the experiment, you're going to compare whatever the response variable is. So you'll be comparing the treatments in regard to what the response was. Another term for outlining an experiment is called making a diagram of an experiment. And you can find examples of diagrams and outlines on page 295 in your textbook and 297 in your book as well. So to wrap up today, see if you can draw an outline for problem 534 on page 293 using the examples on page 295 and 297. Check your answer with your teacher after you finish.